only me in the meeting. All right, so we're finishing up this chapter, or hoping to finish up this chapter. I want to go over how to create a map, a genetic map, with a 3-point test cross. Okay, that's really what you need to come away from this chapter with as far as crosses go, as far as doing a map. 3-point versus a 2-point test cross. We left off talking about map units and the percent recombination is converted to this fictional, or I don't know if fictional is the right word, but this made-up unit called a map unit or a centimorgan. And they're, again, just like it says here, they're relative distances, not exact distances, because they're just pretend units. When you see SCO versus DCO, that means single crossover event versus a double crossover event. And actually, the chance, I think we might have mentioned this last time we were together, the chance of having a single crossover is higher than having a double crossover between two genes. And then less chance of a triple crossover, and so forth and so on. And again, of course, crossover is between two non-sisters. That's very important, and that's how you get genetic recombination. So let's just look at genes A and B and look at the single crossover between the two genes, or single crossover on the chromosome. All right, so in this example, the crossover is after gene A and B. So what we see is that there is no change in the possibilities, the possible combinations for the gametes. In this example, gene A and B are far enough apart that the chance of a single crossover between them has been increased, and now we see that single crossover does make a change in the gametes. So 2 of them are recombinant, while 2 of them are parental. Okay, so in both situations, we have a single crossover event, but based on the 2 genes that you're looking at and their location, there is not, or there is, a change in the order or a recombination. The chance of having a single crossover between A and B in this first example is small because the distance between the 2 is small. They're very close together. So the chance of having a single crossover is slight. Okay, so that's an example. What is the experiment? We all know now that crossover occurs in prophase 1. But what is the experiment that proves this? I'm really big on these experiments that are proving things, right? So this is the experiment with the bread mole, the Neurospora crassa. And with this bread mole, what happens is when their little seeds or their ascus in the pod, the way those little seeds line up in the little pod is exactly how they line up at the metaphase plate. So the exact order in the little pod can tell us what happened before, if there was crossover and such. So how this works is, for this experiment, they use these 2 different mating types, mating type big A, mating type small a. And they are nutritional mutants. Mating type big A cannot make methionine. So methionine has to be added to the broth for it to grow up, or the nutrients, agar. Type little a cannot make histidine. So that's the difference between the 2 mating types. They're nutritional mutants and they differ. Okay? And both the methionine and the histidine genes are on the same chromosome. Okay? So what happens is, even though this bread mole is haploid, well, it makes the spores are haploid. So what happens is they, for a short period of time, like you would see when DNA is replicated, they actually, from haploid, make a diploid nucleus. So for a short time, they're diploid. And then they wind up having the haploid spores. So let's look at what happens. So this is what they look like, mating type big A, mating type small a. Okay? So when they come together and the DNA replicates, they have a small series of time when it's actually diploid before it becomes haploid again. And this is exactly how they line up in the plate, and this is how they line up in the ascus. And each one of these little spores represents a haploid number. Okay? So if there is no recombination at all, we would see 4 parental big A phenotypes / genotypes and 4 parental small a genotypes / phenotypes. That's what we would expect. And that's how they actually line up that way. So here are the possibilities that could happen. Can you see this? Is this too small for you? It's small. And how did we say it make it bigger? The zoom thing. Zoom in. Oh, just like that's not a zoom. Every time I have to. Okay. Is that okay? Even though you can't read it. So this is exactly what we're talking. Let's see if I can write this. No? Give up the ability to write when you zoom. Okay, so this is what we're talking about. We have the mating type big A, the mating type little a. I know it's a little blurry, but the DNA replicates, right? And if there's absolutely no crossover, then the big A goes to 1, the big A goes here, the little a here, and then they divide again. And you have 4 big A, 4 little a spores in the ascus. Okay, no recombination. That's one, no crossover, that's one option. Then we can have crossover between a big A strand and a little a strand that are right next to each other. This is what we're most familiar with. This is how we draw it on the board all the time. And when there's a crossover here, now we get big A, little a, and after they replicate, 2 big A's, 2 little a's, 2 big A's, 2 little a's. See that? Just take some time to look at it. It's not something to memorize. It's just something so that you can see the possibilities. Now, another choice is the further away the outer, let's say outer big A chromatid and the outer small a could crisscross, could have a crossover. The furthest from each other could actually have a crossover. And you can see here, now we have little a big, and they line up in that way. Or we could have the inner and the outer, inner big A, outer little a, jeepers creepers, or the outer big A and the inner little a. These are all possibilities. But either way, they're going to line up in this ascus in the exact order they line up in the metaphase plate. Okay? So what they found out was, let's go back one. These are the options. No crossover, crossover. They found out that crossover occurs at the tetrad stage of prophase 1. It is reciprocal with equal chromosome exchange. Okay? This experiment is the evidence to show us that it is during prophase 1 that there is a crossover. Okay? Facts that you hold true. Any questions? That's, again, a single crossover. You already know this. Here's an example of that single crossover. 2 are going to be parental, and 2 are going to be recombinant. So it's you get 50-50. Okay. All right. In order to really look at a genetic map, to make a genetic map, we usually need to map more than 2 genes at a time. And the reason is, my next picture. Yeah, the reason is, if you're mapping just 2 genes, gene A and B, we could have a double crossover between the 2 genes, because if there's a large distance between those genes, and as a result, it would appear as if there was no crossover. And if there is no crossover, no recombination, that would lead us to think that the genes were not linked. So remember I told you if you write DCO, that means a double crossover. And so we usually have to have at least 3 genes to have an accurate genetic map. Okay? So I'll just show you this picture. Here we have big A, big B, big C, big, small b, small a, small c, and so forth. And if there's a double crossover between A and B, and then between, because there's 3 genes between B and C, then we'll notice that the gene in the middle has flipped its position. Okay, I need you to take a little bit of time to look at that. Okay, so again, we started with big, 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 small, 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 small. And if there is a double crossover, the gene in the middle is always going to be flipped. So look at the parental, big, 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 small, small, small. Look at this one. Small, big, small. Big, small, big. So that tells us, even though we do realize it, that gene B is between A and C. Whenever you have a double crossover event, we can find the gene in the middle by seeing which gametes have one difference between the parent, and that difference will be the gene in the middle. Okay, visual, is it good? Gavin? Okay, I can't tell you. I already told you that a double crossover occurs less than in frequency than a single crossover. All right, so to do a 3-point map, genetic map, there's a couple of criteria that have to happen in order to accurately map. First, one parent has to be heterozygous for all 3 genes. Basically what you're doing is a 3-point test cross. One parent is homozygous recessive and the other parent is heterozygous. And in that situation, you'll be able usually to map the genes. Also, it's very important that you have a significant number of offspring. You can't do this with a small n, a small number of offspring, because it's just not, you're not going to be able to see the differences between the different phenotypic classes if you don't do that. And so that's another thing. One thing that students tend to do a lot of times when they're trying to do a problem like this, like you can see, like look at the classes up there. Number observed, 4,600, 4,700. This one only has 3. A lot of times people forget about the classes that have small numbers in them because they think they're insignificant, but they're highly significant with making a genetic map, the small numbers. And so if you look, it's 3 offspring out of the thousands that were, like the 9,000 the total. If you just did 7,000 offspring, you might have missed these 3, and then you would not be able to create an accurate map. So you really need a large number. This is the example in your book on how to do a map. And I like it, but I love my example more. So I put both examples up here. I could double whammy you, but I'm not going to do that. I'm going to show you this and then move on to my example. But you can see first that we had, this happened to be a sex link. So the male happens to be wild type for all 3 traits, see, plus, plus, plus. And the female just happens to be homozygous recessive. Okay? So these are parental. So you cross them together. Since she's homozygous recessive, the only possibility, she only has one possibility for her eggs. He, as far as X and Y, his Y is this, and then this is his X. So we know for a fact that the sons are all going to be homozygous, or rather, they're hemizygous recessive, and the daughters are all going to be heterozygous. So now you've crossed a brother and a sister. You're hoping it's a fly and not a human, a brother and a sister. And now this is a test cross, basically, a heterozygous crossed with a homozygous recessive. And a map can be made this way. So let me just see if I can, if you don't mind, I'm going to go to my notes. Actually, let's take a second to look at this possibility before we get there. Here are these 3 genes PR, V, and VM. All right? And in this example, they're trying to figure out what is the exact order of these genes and what are the map units between each gene. So if you're just going to cross these genes A, B, and C, and figure out we want to do a map with them, you still, just because I'm telling you, look at genes A, B, and C, we still don't know what gene is in the middle, right? So here are the parentals, right here. Okay? And now start looking at, and I'm right with that, those are the parentals, yeah, non-crossover. So start looking at the offspring and see which ones are different than the friend. This couldn't be right. These are the possible arrangements of real variables. Okay, yeah, this should have been the parental. So what's different than here? So here we have plus VBF. So now we have plus BF. PRV plus plus. So which one is different than the parental? The BM. Am I right? No. The V. The V. Right? The V is in the wrong direction. Anything? Bueller? Yeah. Okay, so because see it's plus VBM, PR plus plus. So the one that's different is the V. It's in the wrong order. I feel like I understand it. Test. Maybe this is not indication of. We'll just go right past that because the actual looks on your faces just not very good. Let's just try this. Boy, I really feel sorry for anyone watching this video in the future. But yet again, 2 days in a row, bad news. We'll just start fresh. Hey, let's make a genetic map. Let's try how we would construct a genetic map. Let's try it. Constructing a genetic map. Okay. Number one, in order to construct a genetic map, the genes of interest must be on the same chromosome, right? Everybody gets that. It's like a basic thing. You can't map the distance between genes A and B when they're on different chromosomes. So first thing, the genes have to be linked, okay? Another thing is the way that you do this, I mentioned just before, we do a test cross, a heterozygous parent crossed with a homozygous recessive. If the genes are not linked, there should be a 1-to-1 ratio of parental and recombinants. If they are linked, there would be deviations from those predicted ratios. That's why, remember, I asked you that you have to remember the predicted Mendelian ratios so that you can see that these ratios are different. Let's move on. Another thing that you need to do to construct a genetic map is to do statistics. We've already talked about the chi-squared analysis, right? We talked about taking the number expected, the number observed, the subtraction, the squaring, and all that, right? And the degrees of freedom. Again, if two genes undergo typical Mendelian genetics, if they are not linked, we should have a 50-50 or a 1-to-1 ratio of parental to recombinants. We would then use the chi-squared analysis to see if the difference between expected and observed is significant. Because if it's not significant, we can't move on. Okay? And usually we're going with a value of hopefully 1%, 5 to 1%. This is repetitive from the very beginning of the course when we talked about chi-square. I'm just going to skip that right now. We know about the map distances. So, I'm skipping, just going to get to something that I feel like we could sink our teeth into. The very first thing that you have to do, I'm sorry if I'm moving backwards, guys. 1 second. The very first thing you have to do when you're doing a genetic map is go in with the null hypothesis. I thought that I wrote this down, but I guess I didn't. I know I wrote it, I just don't know where it's coming up. Maybe here. Yeah, null hypothesis. So you want to map genes A and B. You're not A, B, and C, let's say, but you're not sure, 100% sure that they're on the same chromosome, the same linkage group. So in order to determine that, before you can move on and map the genes, you have to do this test cross utilizing the null hypothesis. So in the null hypothesis, you'd say that the genes are not linked. It's like opposite of what you want it to be. The genes undergo normal, typical Mendelian genetics. What was that?
The 1-to-1 ratio.
And they would have, and we expect, a 1-to-1 ratio of parental to recombinants. So the first thing you do with mapping these three genes, right, is determine if they're linked or not. And to do that, you have to do a test cross utilizing the null hypothesis, that the genes undergo independent assortment, typical Mendelian genetics, they're not linked. Okay? You're going to cross a heterozygote with a homozygous recessive. And you should see a 1-to-1 ratio of parental to recombinants. And you could use the chi-squared analysis to determine if the differences between observed and expected are significant. If there turns out to be a significant difference, then that means that the hypothesis is rejected, that the genes do not sort independently, and they are linked, and you can move forward. But if you do not see a statistical difference between observed and expected, then it means that the null hypothesis is accepted, the genes undergo independent assortment, and you cannot move forward. So really what you want is to disprove your hypothesis. You're going into with the hypothesis saying that the genes undergo independent assortment. You want to find out that there's a significant difference between observed and expected, and that they don't sort independently, which would mean they're linked, and you could proceed. Does that make sense? Yes? Josh, it does? Gavin? I said, does it make sense, Gavin?
Yeah.
Yeah.
Can you go over and do the full one?
Yeah, I'll do one, yeah. But this concept. Yeah?
What's considered the significant, and that's part of this?
You use this formula, chi-squared, observed, expected, the difference between them, then divide it and sum it together. Remember you looked on that chart, didn't I show you that chart with degrees of freedom, which is the number of phenotypic classes minus 1, and you look at the percentages, you find the number, and you see. And you want
a chart.
Yeah, there's a chart to look that up. You're not going to have to do that, unless that's your job. And then there's a computer program that's going to do that for you, anyway. But you could look it up, and then you would like to have a significant difference between observed and expected. That's 1% or less. And that means that the hypothesis that the genes are not linked, or that the genes sort independent, is rejected, which means that the genes are linked and you can move forward. But if there is not a significant difference, you have to stop and say, I'm sorry, I cannot map genes A, B, and C because they sort independent. They're not on the same chromosome. All right? So let's assume you have this null hypothesis. You did your test cross. You did the chi-squared analysis. And you saw there was a significant difference between observed and expected. So then you can move forward. Okay? That has to be the situation. So let's go to this example. I like this example because I don't know why I like it. I just like it. It has a picture of the fruit being purple and juicy, and I don't like the picture. It's stupid. But anyway, when you're crossing these genes, in this example, it's not A, B, and C. It's P, R, and J. Okay? When you're crossing them, here's the homozygous recessive, and then plus, 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 P, J, R is the heterozygote. When you have 3 genes, it's 2 to the n, where n is the number of genes, pairs that you're looking at. So there will be 8 phenotypic classes expected. Right? So you know that there's going to be 8 phenotypic classes. So the first thing you need to do, or it's not at this point, is to write out all 8 phenotypic classes.
The N is the pairs?
N is the number of genes that we're talking about. Gene pairs, yep, genes. So there's 3 genes, P, R, and J. So there are 8 possibilities. So you could have like a purple, round, and juicy. Or you could have a purple, round, and dry. I don't know. These are purple, round, and juicy. Purple, round, and dry. Or you could have a purple, well, it's not round, it's long and juicy. Or you could have a purple, long, and dry, right? So you write down all the possibilities, to which there are 8. And then, after you write them down, you put the exact number, not a percentage, the exact number of offspring that you have of each of those classes down in your chart. So here's your chart, right? So you had whatever, yellow, elongated, dry, purple, round, and juicy. And you put the exact numbers. This is a typo where these all went together and you could spread out. But if you could, I can rewrite it. Then, yellow, round, juicy, purple, elongated, dry. You write down all 8 classes and the exact number of offspring in each one. So for a second, I'm just going to do that horrible teacher thing that you all hate. I'm going to go back to this other example that's from your book, and they did the same exact thing. They have the 8 classes right here. They didn't write them out like purple, round, and juicy, but here they are, the 8 classes, and they wrote the exact number of each one. See that? That's all they did. But it's just more confusing, this chart, I think. That's why I'm choosing to go with the purple, round, juicy. It's a little bit easier for my small mind to grasp. Okay. So when you look at this, you can see it's obvious. Your eyes are going to the classes that are parental and have the largest number of offspring, to the classes, and then all of these are recombinants. There's only 2 parental classes. And then all of these recombinants, these 2 have the smallest number of offspring. So what we can assume from this, we know the parentals, because we know purple, round, whatever it is, purple, round, juicy, dry, elongated, and yellow. And the 2 phenotypic classes with the smallest number of offspring, these represent the double crossover events. Wow, that's not working. It's not working, my pen. A double crossover. And then these 4 here are single crossover events. Pen is not writing very well. Okay. I feel like I'm writing in Greek. Yeah, that's true. So parental, double crossover. So then the key here is to look at the double crossovers and see which order is different than the parental. And this can tell us the gene in the middle. So we have a purple, why is this all of a sudden not working? Round, juicy, right? And then we had a wild type, wild type, wild type. That's what it was here. Wild, 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 purple, round, juicy. Here we have yellow, elongated, juicy, purple, purple, round, wild type. You see how I did that? Right? So which gene is in the middle? What's the correct order? All what? Just use the letter, like P, whatever, What gene is in the middle? What gene in the double crossover is in a different order than the parental? You're comparing these 4 to these 4. What did you say, girl? J. J is in a different order. Now, does everyone see that? Yeah, that's all it is. Compare this plus, 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 PRJ to plus, plus a J, PR plus. The gene in the middle, the gene that's in a different order is gene J. So that means this is truly the gene in the order. So now we have to rewrite the cross in the correct order, which is not the cross of PRJ, but the right order is PJR, right? Cross with PJR. I'm sorry that all of a sudden this pen is like crazy writing. Yes, it can, very good. It can also be RJP. We don't know which side is which, but we do know that J is between P and R. Right. You have to actually just pick something to start with, and that's a random. But see, that's pretty easy. Once
a lot of steps, but
it's a lot of steps, but and after a while, like if you do problems like in the back of your book or just go problems online, they'll list out that like they usually do a triple cross, but they can do a larger cross, 4 or even 5. You'll see the parentals, you'll see the double crossovers, and you'll just start looking and you'll see which gene is in a different position. And it'll be difficult when more than 3, because with 3 it's actually the order where 1 is directly in the middle. But you'll be able to tell, usually your eyes will go right to which gene is in a different order, and you'll be able to write the cross in the correct order.
This could be an essay test.
This could be an essay question. Yeah, I can go over it again for sure. You know? So now you have figured out that the right order is PJR. Okay? Because if you went back and you were trying to figure out the order and it was and the map between PRJ, it would never work for you. You could not get it to work. So now what do you do? Now the total number of offspring, in this example they made it easy, is 500. Right? So now you have to figure out the percent recombination in each region between P and J and between J and R. And you can also do it if you want between P and R. That should be additive. So the first region would be P to J. The second region would be J to R. So you need to figure out the percent of single crossovers in each region. This is region 1 and this is region 2. Percent of single crossovers, and also the percent of double crossovers, because the double crossovers go for each region. I don't know if I have a slide of it where they write it out. No. Yeah. So again, they have now we've gone from P to J is region 1, J to R is region 2. Okay? And here are the numbers. So I don't know if there's math on the next page. No. So the single crossovers, look here, plus JR, P plus plus. This is a crossover between P and J, right? Plus plus R, PJ plus. This is a crossover in the second region. You see that? This is no crossover. And this one is a double crossover. So you add together for region 1, 52, 46, and then 4 and 2, the single crossovers and the double crossovers. And for region 2, you add together 22, 22, 4, and 2. And if you add that up, like it's 10, 14, 104, 50. Okay, so now we have the number. If I added right, I think I did. This is the percent recombination in between P and J, percent recombination between J, or actually not percent, the number between J and R. Then divide this by 500 to get your percent. And that will be the distance between P and J and R. So our map turns out to be 20.8 and 10. So 104 divided by 500 is 20. Look at you, Gavin. You're like, oh, well, I have to do this. And you can make that map. I think I've looked at a lot of problems in the back of the book, and maybe not this book, but my old book, and I can just see how to do it. What do you think? You're okay with it, Chris? I can tell. Josh? Easy, right? Math-minded people probably get it really fast. What about you, Sophia? Good? Still? I just run through it and just talk to you about it, and then see if it makes sense when I talk to you about it without giving you an example. I can keep this up, all right? So the first thing you have to do if you want to map these genes A, B, C, PJR, whatever, is you need to find out that those genes are actually on the same chromosome, right? That they're linked, because you cannot map them if they're not linked. To figure out if they're linked, you're going to utilize the null hypothesis. And you're going to say, these genes are not linked. They sort independent of each other, right? And so to test your hypothesis, you're going to do a 3-point test cross. A heterozygous parent with a homozygous recessive. You're going to use the chi-squared analysis. I mean, you're not really going to do this problem. You would just be writing out the steps that you would. This is like an essay of writing out the steps. You know what I'm saying? I'm not going to ask you to really map anything, okay? Take it back. No. I want you to just understand the steps. So if I give you numbers, you could do it, but I'm not giving you numbers. I just want you to understand the steps. So the first step is does that make you feel better? First step's the null hypothesis. Do the 3-point test cross. Use the chi-squared analysis to see if there is statistical difference between observed and expected. If there is not a statistical difference, that means that your hypothesis is rejected, is accepted, I take that back, and the genes sort independent, and you must stop. You cannot go any further. But if there is a statistical difference between observed and expected, then you can continue with your mapping, because that means that the genes are linked. They're on the same chromosome, the same linkage group. So then you need to write out the 8 phenotypic classes. In the case of a 3-point test cross, there will be 8. You write out the exact 8 classes and put the exact number, not percentage, of offspring in each of those 8 classes. Then you can obviously know the parental, because they were exactly what the parents look like. They should have the highest number. And then see the groups with the 2 groups with the smallest number, which represents the double crossover event. By looking at the phenotype of the double crossovers and comparing it to the parental, we can tell which gene is in the middle. And if necessary, we rewrite the cross with the correct order. We might have been lucky and we had the right order from the beginning, but we might not have. Logan, okay? Yeah? And then you figure out the percent single crossovers in each region, and then also add the double crossovers to get the frequency of and then divide by total to get the frequency of recombination. Take percent or frequency of recombination and convert that to map units. That's how you do a cross. Or that's how you figure out a map. But you can't do it if you don't have a heterozygous parent. You know what I'm saying? You can't do it if they're all the same. Good? You feel good about it? Like you can pray that's on there because you're going to write those steps out. That's going to be good.
We talk about converting the map unit from this point. Let's go with that.
That's all you do. It's like the percentage 20%, 20.8%. You just go 20.8 map units or centimorgans. Yeah, that's really all it is. Crazy, right? I mean, that's why it's relative. It's not really like a map that's been determined. I've actually had some really good. I know I'm going over here, I'm going all over the place, but some offspring have been really mapped. Like here's the fruit fly, and you can see how it's been really, been mapped. But this is just, if you just happen to look at this picture just for a quick second, when they're first, like chromosome 1 here, when they're first mapping it, this is actually the X chromosome, just like Brianna said, is the yellow body the first gene on this chromosome, or is little fly the first gene on the chromosome? It depends. In the beginning, if you're mapping yellow body and little fly, those 2 genes, you wouldn't know which one goes which arm. You're just randomly putting one up at the top and one up at the bottom and hoping you're eventually going to figure that out, which they obviously did. So crazy. So here's just an example. Very well looked at. Okay. So how are we doing on time? Well, I know I went all over the place here. I just want to make sure I hit all the bits of information. I kind of wrote all of this out and highlighted it for you about how to do the map. One of the things that's very interesting is this little statement right here. That in reality, they're typically these recombination frequencies, which we convert to map units, is not really the truth. Because once you have a single crossover on part of a chromosome between, let's say, genes A and B, it decreases the chance of having a second crossover. And that's called interference. And so even though they make up a map utilizing this technique, they have to take into account that it can be inaccurate, and there can be what's called interference. And I am not expecting you to be able to utilize interference. I just want you to be aware that interference means that further crossover events may not occur. A nearby crossover event could inhibit a crossover event between the genes you're looking at. And then there might be a decreased frequency of double crossover or multiple crossover events. So they actually have a formula: observed double crossovers divided by expected crossovers, and then take that value and minus it from 1, and that gives you your interference value. And so if you have could have a complete interference when no double crossovers occur. If the number is positive, there it means fewer double crossovers occurred than expected, and if the number is negative, more double crossovers occurred than negative. These are not going to be vocabulary words, even though they're highlighted. I just wanted to bring to your attention that we can do all the math we want from these classes, but the reality is the chromosomes may not be cooperating, and we might be underestimating the situation or overestimating the situation. Okay, so they actually realize that, so they have that. And you can see what happens here when you look at map distance, your units, and percent recombination. In theory, there's a 1-to-1 ratio. If it's 10% recombinant, it's 10 map units. In theory, it's a nice straight line, right? 50-50. But actually, as you get further away, it's decreasing. And that's due to interference. So that helps us remember. And another thing that's very interesting, if you just happen to look at this 2-strand double crossover. So you're starting off big A, big B, small, 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 small. There's a double crossover between these two genes. Guess what? It looks as if there's been no crossover. There's nothing detectable because the difference between the genes is so far apart. So you may do a chi-squared analysis, and it appears that observed and expected are the same, and you might agree that your hypothesis is true, but the reality is the genes are so far apart, they appear as if they are unlinked, as if they are on 2 different chromosomes. So that's why we always need a gene in the middle. We need to map smaller units, because if there are bigger units and there are even number of crossovers between the 2 genes, it appears as if there's no crossover at all. So that's why if you were just doing 2 genes, a 2-point test cross, now this is working fine. 2-point test cross is less accurate than a 3-point test cross. And that's because the distance between the 2 genes may be so large that there are multiple crossovers. So if, I just want to write this down because, to be honest with you, sometimes I give this question on the final. If distance between 2 genes is far apart, then there can be multiple crossovers, and if the number is an even number, it can appear that, well, here, the genes are not linked. So a 3-point test cross is going to utilize smaller distances on the chromosomes. This is so sloppy, I'm so sorry. I'll say it again because I'm sure you can't read it. On the chromosomes between the genes. So I wrote, a 2-point test cross is less accurate than a 3-point test cross. That's like a fact. Why? Because if the distance between 2 genes is far apart, like it was for gene A and B, very far apart, then there can be multiple crossovers between those 2 genes, and it can appear, I should say, it can appear as if, but it can appear that the genes are not linked. So a 3-point test cross utilizes smaller distances on the chromosome between the genes. So you'd put a gene C here. So you'd look the distance between A and C, and then C and B. And that smaller distance, the chance of a double crossover is less. Does that make sense to you, Andrea? It's hard to read my handwriting. I totally respect that. I can't even read that. It's way easier, you think, to write on this thing than reality. between the genes. Abbreviation BTW, between the genes. Okay? So write out the steps on how to do a genetic map, like I just talked to you about now. Okay? You don't have to use real numbers, it's the concept. Okay? Take a picture. Take a picture, try to decipher that at a later date. Okay. So we just have to finish up this chapter next time, and that's Tuesday, a week from today. And what else can I tell you? Well, we're going to start with, yeah, I have it up on my computer. We are going to start next, I don't know if we'll start it on Thursday, but we will be starting mapping in prokaryotic cells, which is, it should be a review if you've had microbiology, and much easier. Not that this is hard. Okay, I'll just finish up this chapter with a little odds and ends and move on to the next thing. I don't have a lot highlighted on this chapter. I love the last slide, though. The last slide is good.
Well, on the actual the structure and folding and that.
No, not protein folding and protein structure. No, no, no. I'm glad you asked. That's a good question.
Post-translational.
Yes. That's what I handed out to you today, that or protein sorting. Another question? Yeah, that's right in your hand. Great. Okay. Other questions? And this for this class, Josh?